Hi, this is Miss Litton, and this 7th period support. Say hi.
Hi.
And we are continuing, we started Big Idea 2 about biological systems utilizing energy and molecular building blocks to grow, to reproduce, and maintain homeostasis. And we started working on Enduring Understanding 2A, about growth, reproduction, and maintaining organization of living systems require energy and matter. We have already reviewed the laws of thermodynamics and delta G, and we already discussed photosynthesis, and now we are moving on to cellular respiration. So the big idea here is that you're, for our purposes, we oxidize sugar and break it down in order to gain ATP from that process. And we do what's called aerobic respiration. But organisms can do anaerobic respiration. But anaerobic respiration, you walk away with such a small amount of ATP, you leave a lot of energy still left in that sugar molecule that we couldn't survive on such a low turnout of ATP. We need to do oxidative phosphorylation, and we do that last step there in our mitochondria. But the first step, which is done by who? All cells. Good. That's huge. You can't say that about everything, an all cells type of a statement. And so you want to pay attention to that idea because that's something that's conserved throughout life processes. And that first step is called glycolysis. And that's where you ultimately take glucose and break it down in how many steps? 10 steps using 10 different enzymes. And your end product is pyruvic acid. But to get that process going, right, you have to invest a little bit of energy to get those bonds breaking. And how much energy does it cost you? 2 ATP. But when you walk away, yay, when you walk away with how many? 4, right? So you net out when you're all said and done with 2 ATP. Now, the next step, it would be transition, not shown in this picture, but it's where it says if O2 was available. And you're going to move in, if you're eukaryotic, you're going to move into the mitochondria. And in that process, you're going to take the pyruvic acid and convert it into something that can be used in the Krebs cycle. And what is that thing that can be used in the Krebs cycle? No. Acetyl-CoA, baby, right there. So you transition into acetyl-CoA. In the process of doing that, you don't make any ATP, but you make 2 more of something you made in glycolysis. And that is, you're reducing something. What are we reducing? NAD. And in the process of glycolysis, you reduce 2 NAD. In the process of transition, you reduce 2 more NAD. And we're going to accrue those NAD in all of these steps. As we oxidize one thing, we always reduce another, right? And so we're going to start to build up our reduced NADs, and those we're going to take to the bank of the ETC so we can make some ATP, right? So we make a couple NAD. Then in the process of the Krebs cycle, you feed it acetyl-CoA, but just like in the Calvin cycle where RUBP was preloaded, you have a 4-carbon compound already preloaded in your Krebs cycle, and that 2-carbon acetyl-CoA hooks up with a 4-carbon, and tell me how it goes. 2 plus 4 is 6. Do the bit, and when we do the bit, what are we doing? Reducing NAD, pulling off a CO2. 2 plus 4 is 6. Do the bit, get 5. What? Do the bit, get 4. Make an ATP, reduce an FAD. Reduce an NAD, and we do it again because we have 2 acetyl-CoAs, right? So again, we generate more NADs and FADs. So you take all of those NADs and FADs to the top of the ETC, where they then become oxidized, a series of electron transport molecules. As they're transporting the electrons at like maybe 3 locations, you have enough energy to generate a hydrogen ion gradient, a chemiosmotic gradient. And you're going to take hydrogen ions from the matrix, right? Reductase, what? Cytochrome oxidase, and put it in the intermembrane space. When that comes back in through ATP synthase complex, you're going to generate ATP. I mean, that's the whole thing in a nutshell. Now, we saw when we do oxidative phosphorylation, you can see how you're regenerating, you're kind of cyclical and making your NADs reduced and then oxidizing it at the electron transport chain. But if you do fermentation without any oxygen, you're still going to have to regenerate your oxidized NAD. And in this case, you either take your pyruvic acids, and you and I, we make what? Lactic acid makes our muscles sore, but plants might convert it to alcohol. Now, the other thing we want to think about is how many carbons are in glucose? Six. So we know where those 6 CO2 the 6 carbons are released as CO2. We know where those occur, right? Tell your bio buddy, where did those occur? Where do we generate the CO2? Did they say transition prep? Remember this? Here we go. And glucose, which is 6 carbons, you break it down into 2 3-carbon molecules, you pull off CO2, right? There's 2 CO2s. Okay, then we do 2 plus 4 is 6, do the bit. There's 3, do the bit, get 4, there's another one, right? 1, 2, 3, 4, and then we're going to do, come on in. A tour, yes, bring your tour in. And then we're going to do it one more time, right? We'll do the Krebs cycle, so we'll get a total of 6 CO2s generated as a byproduct of that, yes? Okay, and I'll stop just momentarily. Hi, just a minute. I'm going to pause you. All right, so I think we feel comfortable with this first part. Can I keep going? Yeah. Can we review that? Okay. What, you can't do this math problem? Okay, and here's the transition. And here we can see just following the 3 carbons, and where did those 3 carbons come from? Glucose, thank you. Broken in half, right? So here I see these 2 carbons, and then here's the 3rd carbon over here, right? Released as CO2. And we slowly oxidize that. Remember, oxidize, you lose energy, reduced, you are gaining energy, right? By gaining hydrogens, gaining electrons. Okay, and then here is your Krebs cycle, which you already know and love. You feel comfortable with that? They're not going to ask you to memorize these steps in the Krebs cycle. They're not. Okay, but they would expect you to understand what's occurring. If you could summarize what was happening, and you know you're continuing to oxidize your glucose, right, in order to generate more reduced NAD. You don't make a lot of ATP here, right? Each turn only gets you 1 ATP. The bank is the mitochondria. Now what you see here, this we see in energy processes a lot, right? We also see it when we're trying to have a cell communicate from one end of the cell to the other cell, even in a neuron, right? These differences in charges, these differences in concentrations. We can use that to have a neuron fire. We can use that to generate a hydrogen ion gradient, so you can generate ATP, whether it's in a chloroplast or a mitochondria, right? So there's some similarity, and this is where you can see the importance of that cell membrane creating a barrier. Because if there's differences, just like a dam, right? Water on this side, no water on that side. So if you let the water go through, you can use that to harvest energy. It's that same repetitive process. Okay, and there are some questions. We want to start them, or you want me to keep going? What do you want to do? Keep going? Yeah. Okay, good deal. Here is anaerobic respiration. So you're starting with glycolysis. That part's the same. But this is when you don't have oxygen, and the strategy here is whatever type of organism it is. So for us, we're going to make lactate, but a plant might make ethanol. Either way, the whole point of fermentation is to do what? Why does fermentation even occur? Yeah, to oxidize that NAD so it can be reused in glycolysis again. Okay, now speaking of energy, if we're going to talk about energy, we want to talk a little bit about food. Okay? Because food, we need the glucose, first of all, in the bodies and in the cells so you can do cellular respiration. So how do you access that glucose? It's going to come from your environment, right? Something's got to come. If you're a plant, you're going to be doing the reactions of photosynthesis to generate glucose inside your cells, right? Inside your chloroplast in order to make that glucose. If you're a heterotroph, you have got to consume organic molecules in order to get access to glucose. Whereas plants take inorganic molecules like CO2 and convert it to glucose. All right, so in heterotrophs, you can do extracellular digestion, and it has the superstar because that's evolutionarily more advanced. we have a tube within a tube system. We throw food in that tube, we throw enzymes in that tube, and we absorb the nutrients out of that tube. Okay, intracellular digestion, you're engulfing through what process? Phagocytosis, good. And then you're hooking up with a lysosome to digest what you've just engulfed. Okay, so every nutrient then has to be digested inside within that cell, whereas what we will do is we'll just throw a whole bunch of things into the tube and then we can absorb a whole bunch of things, right? Either through diffusion or through active transport. Okay, we talked about body fluid regulation and how things can get in and out of our bodies, areas where we have exchange, like in our, how do we exchange gases? For you and I, where do we exchange our gases? In the lungs, right? Alveoli. And we have to have moist environments because we need oxygen for oxidative phosphorylation, right? So that's how we're going to acquire our oxygen. We're going to need water to maintain homeostasis. So we're going to get that through digestion. So there are places where we interact with our environment. And then that brings us to the 4 important organic molecules that we could get. And you would want to know, these are the walls, right? So you would want to know the monomers of each of these. Do you remember the monomers? What are the monomers of carbohydrates? What? Monosaccharides are sugars. How about lipids? There's 2 categories of lipids.
Steroids.
Fats and steroids. Steroids are made out of 4 rings of carbon, and fats are glycerol and fatty acid chains. Those fatty acid chains could be unsaturated. Animals tend to have saturated. Boom, shakalaka. Yes. Proteins are made out of amino acids. How many different amino acids? 20. Do you remember the structure of an amino acid? Yeah. Central carbon, hydrogen, an R group, a carboxyl group. I'm trying to make an R. A carboxyl group and a amino group, right? Okay, and nucleic acids are built out of nucleotides. What do you call the reactions by which you join them together?
Dehydration synthesis.
Perfect. Dehydration synthesis. Another name? Condensation synthesis. How about when you're breaking them apart? Hydrolysis. Because you have to have the input of water to break them apart, right? So this is not the nice diagram. This is my messy diagram. But I gave you a nice diagram. Remember that? Yes.
With hydrolysis, so water's being put in, and then you're having 2 products forming as like water.
Oxygen will go to one, and hydrogen will go to another.
So if I have like,
okay, stop and just listen for a minute. Okay. So if there is a bond, okay, reach out your hand. Okay, if we're in a bond, right? So we're sharing a pair of electrons here. We're happy. So if you're going to try to break us apart, you need to give something to fill the gap because I don't have the electrons I need. And that's I'm going to use water. An OH for one of us and an H for the other. Does that make sense? Okay. Then that brings us to talking about the different, and this is at the very end, so we're almost done here. This brings us to cycles. Now, we haven't talked about these cycles yet, but I know you talked about them last year, right? So this should be a review. So what cycle is this right here? Right, right. So what do you call rain? What's the name for rain? And what do you call evaporation out of a plant? You've already learned this. Transpiration, right? So we know about the water cycle, okay? And what's this cycle? Phosphorus. Yeah, you've learned it, right? So phosphorus, so when might I want phosphorus? Adenosine triphosphate? Yeah. How about this? In DNA, base sugar phosphate, right? So phosphates are important, right? How about this one? Oh no, not that one. Okay. What came to mind was really stupid.
Actually large, just
yeah. He's a chocolate bunny. All right. So what you're going to find out in phosphorus is that the reservoir for phosphorus is in the rocks, and then through erosion and weathering, it puts it into water. It's a limiting factor. It usually gets used right away. So then it moves into the organic community. It'll be in plants, and then animals can eat plants. Plants and animals can what? Die, and then decomposers work on them, and it gets back into the system. All right. Then another cycle we're going to learn about is the nitrogen cycle, and I'll review this at the time, but I want to tell you this. Do this. Put your hands in the air, pre-teaching. Okay? Nitrogen fixation. Go grab some nitrogen. Nitrogen fixation. The reservoir, oh, we're cleaning right now, by the way, just cleaning. The reservoir is N2 in the air, and then fixation, you fix it into ammonia, NH3. You do nitrification, forming nitrate, nitrite, NO2 minus, and then more nitrification by bacteria to form nitrate, NO3 minus. Nitrogen fixation and nitrification, forming nitrite, nitrate, all done by bacteria. By sweetheart. If we don't have the bacteria, we gotta have it. Then plants can eat and use the nitrate, and animals can eat the plants, and then animals and plants die. Who works on us? Decomposers, and it goes back to ammonia. Whole thing happens all over again. Moving through living systems. You can get it out of living systems and back into the atmosphere through denitrification. Puts it back into N2. There's also atmospheric fixation where lightning does that. I'm giving this in this right here just in case somebody watches this review after we have learned this. I wanted to review that process. Okay, and then last but not least, carbon cycle. And we've reviewed that already, and that we've talked about cellular respiration and photosynthesis. But what's the big concern on this cycle? What? Yeah, burning fossil fuels, right? All of these man can screw up, right? Fossil fuels. If you look here in the nitrogen cycle, again, pollution. When you look at phosphates, if you've heard of eutrophication, or soaps, when they give you the phosphates, so they'll say these soaps, like if you're going to go camping, don't have phosphates because it can cause an algal bloom in a lake. And then we can pollute our water cycle. So all of these have the ability to be destroyed by man. Okay, and then we can pick up next time at 2B. Good job, guys. Way to review.